Good morning, class, and welcome to week 11, neural tissue. We just did our whirlwind 2 weeks on the muscular system, and today starts our last system for this semester, which is the nervous system. The nervous system has a lot of different functions. It directs immediate responses to stimuli, and the word immediate here is very important because the endocrine system directs long-term changes, but the nervous system directs immediate responses to stimuli. It coordinates and moderates activities of other organ systems. The nervous system is really in charge. It's like the mainframe computer, and it tells everything else in your body what to do. It provides and interprets sensory information, so you use it to sense your outside environment and then send those sensations to your brain for interpretation. And it works to maintain homeostasis along with the endocrine system. In addition, it's responsible for our perceptions, our behaviors, our memories, all of our higher order functions, and it initiates voluntary movements. The major structures involved include the brain and spinal cord, our nerves, so our peripheral nerves, and our sense organs. Those are the parts of our nervous system that allow us to sense pain and temperature and texture and all of those different things. We have two different cell types, two main categories, and then the second cell type we're going to break down. So our neurons are the basic functional cells of the nervous system. They are specialized cells for communication. I would remember that. Neurons are specialized for communication. And then our neuroglia is a group of multiple different types of cells that act to support our neurons. So neurons are the main functional unit, and then the neuroglia are our supportive cells. Our nervous system is broken into two divisions. The central nervous system, which includes the brain and spinal cord. This is responsible for integrating, processing, and coordinating sensory data with motor commands. So sensory data comes into the brain and spinal cord, and motor commands are issued in the brain and spinal cord and then sent to the muscles that need to move. And our brain is also the seat of higher intelligence, memory, learning, emotions, all of those higher functions that our peripheral nervous system cannot do. So the second division is the peripheral nervous system, which includes everything that is not included in the brain and spinal cord. So all of the neural tissue outside of the brain and spinal cord is considered peripheral nervous system. Peripheral nervous system delivers sensory information from the peripheral tissues to the central nervous system and and then it carries motor commands from the central nervous system out to the periphery. Nerves are bundles of axons, are the nerve fibers that carry sensory information and motor commands in the peripheral nervous system. So we don't have nerves in the central nervous system. We have neurons and tracts that make up the brain and the spinal cord. But in the peripheral nervous system, our neurons form bundles that make up nerves. We have two types of nerves, cranial nerves, which are connected directly to the brain, and spinal nerves, which come off of the spinal cord. The afferent division carries sensory information from the peripheral nervous system to the central nervous system. So afferent is bringing information toward the central nervous system. And then our efferent division carries motor commands from the central nervous system out to the peripheral nervous system. So it tells our muscles to contract and our glands to secrete. receptors and effectors within the system. So receptors are focused on detecting change. So they're detecting changes when you have a stimulus. If you're like, hey, that's cold, or that's hot, or that's sharp, those are all detected by receptors. They're made up of neurons and some specialized cells. And we have some complex ones in the eyes and the ears that are complex sensory organs. And you're going to learn more about those in A&P 2. But for now, we're just looking at receptors are detecting stimuli, and effectors are responding to those signals that the receptors have detected. I know that was a lot of words all in a row. So say you step on a tack. Your receptor is going to feel there's a sharp tack in my foot. And then your effector is going to be your muscles contracting to lift your foot up off of that tack. We talked about the afferent division bringing information to the central nervous system, and the efferent bringing commands away from the central nervous system. But the efferent division also has two subdivisions. So there is the somatic nervous system, which controls skeletal muscle contractions, both voluntary and involuntary muscle contractions. So the somatic nervous system, skeletal muscle. That's how you can remember it. S for somatic, S for skeletal muscle. The autonomic nervous system controls everything else. So subconscious actions, contractions of smooth muscle, cardiac muscle, glandular secretions, and the autonomic nervous system is broken down into the sympathetic division, which has our fight or flight effect, and we're going to talk more about that later. And the parasympathetic division, which has more of a relaxing effect, we call that our rest and digest. So these two are going to come up over and over again for the rest of A&P 1 and A&P 2. So sympathetic is fight or flight, parasympathetic, rest and digest. So let's break it down to the smallest units of the nervous system. So we're going to talk about our neurons first. Multipolar neurons, when we did tissues, we talked about nervous tissue and we looked at a multipolar neuron. This is the most common type of neuron in the central nervous system. It has a cell body, which is here. All in blue there is the cell body. And that has a nucleus surrounded by cytoplasm and includes your traditional organelles. The dendrites, which are here, look like little trees coming off of the cell body. Those are receptors, and they are looking to receive information from other cells. So whether they're receiving it from other neurons or other sensory parts of the sensory receptors, dendrites are receiving information. And then the axon is this part here, and the axon is a long, thin, cylindrical projection that propagates the nerve signal toward the next tissue. So the propagates means carries along. So the information coming into the neuron travels in a specific way. So we have the information comes into the dendrites, moves through the cell body, and then out the axon to whatever this next cell is going to be. So that next cell could be another neuron, it could be a muscle cell, it could be a gland. But this is the flow of information. The dendrites are receiving the information, then that signal gets transmitted through the body, and if it creates an action potential, which we'll talk about later, it's going to propagate down the axon. So a couple weeks ago, we looked at the neuromuscular junction. And so the neuromuscular junction is right here. So this is where the acetylcholine was getting released into the synaptic cleft, going to bind to the receptors on the sarcolemma. So that all takes place right here at the end of the axon. So we'll look at each of these areas more closely. The cell body has a large nucleus, the nucleolus. The cytoplasm, for some reason, has its own name. It's called perikaryon within the nervous system. So that's the cytoplasm of the cell body. It's got mitochondria to produce energy. It has clusters of rough endoplasmic reticulum and ribosomes, and those clusters are called Nissl bodies, which you can see up there on the right. So Nissl bodies are clusters of rough endoplasmic reticulum and free ribosomes. The cytoskeleton is made up of neurofilaments and neurotubules, which would be the same as microfilaments and microtubules in our traditional cell anatomy. And then neurofibrils are bundles of neurofilaments that provide support for the dendrites and the axon. So takeaway from this slide, I really want you to know perikaryon is the cytoplasm. The Nissl bodies are rough endoplasmic reticulum and ribosomes, and that we do have a large nucleus and a nucleoli and multiple mitochondria, and that there is a cytoskeleton. All right, well, there's our Nissl bodies. I do this every semester. So we already talked about Nissl bodies. Those are what make neural tissue appear gray. We do have a gray look to our neural tissue. Our dendrites off the cell body are highly branched. They have lots of different branches with fine processes on the end, and those are receiving information from other neurons. And then our axon is a single long extension that carries the electrical signal from the cell body to the target cell. So, and we've discussed that target cell can be another nerve, it can be, or another neuron, it can be a muscle cell, it can be a gland. So whatever requires the signal that that cell is going to produce, the axon is going to go and synapse upon the next cell. Within the axon, we have the axoplasm, which is the cytoplasm of the axon. So the perikaryon is the cytoplasm of the cell body. The axoplasm is the cytoplasm of the axon. It also contains neurotubules, neurofibrils, and some enzymes and organelles within the axoplasm itself. And then the axolemma is the cell membrane that covers the axon and surrounds the axoplasm. The axon is a cytoplasmic extension of the neuron, and it can be up to 1 meter in length. That's 3 feet long, the axon can be, which is pretty incredible. The axon hillock is a thick section of the cell body where it attaches to the initial segment of the axon. So the cell body, I think we'll come up with a picture in another couple slides, and I will show you and discuss what the initial segment and the axon hillock are. It's easier with a picture. Collaterals, you can have branches that come off of an axon. Those would be called collaterals. Telodendria are the tips of the axon at the end where it's going to be synapsing on that next cell. So when we were looking at the neuromuscular junction, the acetylcholine was being released by the telodendria, and well, at the synaptic terminal. So the synaptic terminal is the very tip of the telodendria. The telodendria is the whole extension off the axon. So let's look at this in a picture. So here we have the entire multipolar neuron. So first we'll start with our dendrites. These are dendritic branches, and the very tips of the dendritic branches that are spiky are called dendritic spines. Within the body, we have our Nissl bodies, which are right there. We have our large nucleus and nucleolus. We have our perikaryon, which would be the cytoplasm within the body. And then we have the axon hillock, which is right here. So this is sort of where the body starts to taper into the axon. That's called the axon hillock. And then the next part, so this initial part of the axon right here is called the initial segment. So the axon hillock connects to the initial segment of the axon. I would remember that. And then going out the axon, we have the axolemma, which would be the cell membrane surrounding the axoplasm. And then at the end, we have the telodendria, which are each of these. So there's one, there's one, there's one. Okay, so the tip of this has 3 telodendria. And then this little tiny knob at the end of the telodendria where it's, that one didn't work out very well, where it's sort of, that still didn't work out well, expanded at the very end of the tip of the telodendria, that is called the synaptic knob or synaptic terminal, either way. The synapse is the site where the neuron communicates its signal to the next cell. So where we talked about the neuromuscular junction, the synapse is this area, oh, let's do it like this, between the area between the synaptic knob and the postsynaptic cell. Okay, so that's the synaptic cleft. So this whole thing is termed the synapse, where these two cells meet. The presynaptic cell is the neuron that is sending the message because it is before the synapse. And the postsynaptic cell is the cell that's receiving the message. So the synaptic cleft is that small gap in between the presynaptic cell and the postsynaptic cell. So one more time. Presynaptic cell, postsynaptic cell, synaptic cleft. The synaptic knob is the expanded area of the axon of the presynaptic neuron. So it's that end of the telodendria, and that is where we have our neurotransmitters. And neurotransmitters are held in synaptic vesicles. So they're little walled off units, vesicles, that contain neurotransmitters. Neurotransmitters are chemical messengers. So in our neuromuscular junction, it was acetylcholine. So acetylcholine is one neurotransmitter. It's actually the most common neurotransmitter in the body. But we have other neurotransmitters as well, like epinephrine, norepinephrine. There's a very long list, and those are the ones I can come up with off the top of my head here. So they're chemical messengers. They're released by the presynaptic cell, and they affect receptors on the postsynaptic membrane. If there are additional neurotransmitters left in the synaptic cleft, they get broken down by enzymes like the acetylcholine esterase broke down our acetylcholine. And then when the enzymes break them down, it breaks them into their component parts, and then those parts can be utilized by the presynaptic cell to be reassembled into new neurotransmitters that can get released again. Our two types of synapses are a neuromuscular junction between a neuron and a muscle that we've already looked at, and a neuroglandular junction between a neuron and a gland. Okay, so now we're going to break down the different structural classes of neurons. So the neurons we've been looking at are the multipolar neuron, which you can see over here. This is a multipolar neuron. But we have other types of neurons as well. So the first one is called an anaxonic neuron. An means without, and so it's without an axon. So we find these in the brain and in our sense organs. So it's basically a cell body with a whole bunch of dendrites. Bipolar neurons are a neuron with a single dendrite and a single axon, and the body is in between. So you can see the dendrite is coming off that end, the axon is coming off this end, and the body is situated right in between the axon and the dendrite. Unipolar neurons are similar to the bipolar neurons, except that they have their cell body is off to the side. So the cell body over here is off to the side. It's not the axon coming out of one side and the dendrite coming out of the other side. It has a single process that splits into dendrite and axon, whereas the bipolar neuron has two separate processes. And then the multipolar neuron has one axon and a lot of dendrites. We have three different functional classes of neurons. Sensory neurons, which are afferent neurons of the peripheral nervous system, meaning they bring sensory information from the periphery to the central nervous system. Motor neurons, which are efferent, they bring motor commands from the central nervous system out to the peripheral nervous system. And then interneurons, which are association neurons, so they help to sort of integrate information when there's a lot of information coming in. Interestingly, interneurons outnumber all other neurons combined. We don't talk a ton about them, but just know that they outnumber all the other neurons combined, which is incredible. So we have a lot of interneurons. Sensory neurons monitor the internal environment within your body. Those are visceral sensory neurons. Remember, the viscera is the term for your innards, your guts, or your viscera, all of your internal organs. So all of those are being monitored at all times because your body has a very tight homeostatic state and we want to stay in that state. So we're always monitoring to make sure that we are where we need to be. They also monitor the external environment. Those are somatic sensory neurons. So that's what's telling you if you're hot or you're cold, or you're touching something smooth or something rough. Those are all somatic sensory neurons. And our sensory neurons are unipolar. And this I definitely want you to know. Sensory neurons are unipolar, and their cell bodies are grouped in sensory ganglia. This is going to become very important. You're going to hear this over the next few weeks, so it's awesome to commit it to memory now. A ganglia is a collection of neuron cell bodies. So if you picture what the unipolar neuron looks like, you've got the cell body here, and then the dendrite here, and the axon here. So you can have a bunch of cell bodies all together in a ganglia like that. These are all cell bodies, if you can't tell. And then their axons are all going to go together in a nerve. So our sensory nerves are formed by groups of axons traveling in the same direction. And then our ganglia is this part, which is the group of neuron cell bodies. And we only find these in the sensory nervous system. For sensory receptors, we have interoceptors, which monitor the internal environment. So those visceral receptors are called interoceptors. They also do our internal taste, our internal senses like taste, deep pressure, and pain. Exteroceptors, which are monitoring our external senses of touch, temperature, and pressure. So that would be a light pressure versus a deep pressure. And distant senses like sight, smell, and hearing, which are all special senses, and we're going to learn more about them in A&P2. And then our proprioceptors monitor the position of our joints and our skeletal muscles in space. So if you have your eyes closed, traditionally I would make you stand up in class at this point. So you could stand up and close your eyes, and if you were to flex your right arm, without looking, you would know that your right fist was up towards your right shoulder. Or if you were to reach behind you with your left arm, you would know where that arm is in space. And the reason is because of our proprioceptors, which are always sensing where we are in space and sending that information to our brain. Motor neurons carry information from the central nervous system to the peripheral nervous system via efferent fibers. So remember, efferent is away from the central nervous system toward the peripheral nervous system. We have two major types of efferent systems, which we talked about before. The somatic nervous system, which is all somatic motor neurons innervating skeletal muscles. So our somatic nervous system is efferent fibers innervating skeletal muscle. And then our autonomic nervous system, which is more of our visceral motor neurons, which innervate all peripheral effectors except for skeletal muscle. So somatic nervous system, skeletal muscle only, autonomic nervous system, everything else. So smooth muscle, cardiac muscle, glandular tissue, adipose tissue, all of those things are innervated. Innervated means the nerve supply would be the innervation. So they're all innervated by the autonomic nervous system. When we're talking about our autonomic nervous system, we do actually have ganglia within the efferent division, but they're still multipolar neurons, not unipolar neurons. So with the autonomic nervous system, the signal travels along the first axon, so it leaves the spinal cord, travels along an axon, and then relays a signal across a synapse within a ganglia, and then the synapse is onto a second neuron, so then the signal travels along the second neuron to reach the effector. So we have two different kinds of fibers. Preganglionic fibers is the term used for axons that are extending from the central nervous system to the autonomic ganglia. Postganglionic fibers are axons going from the ganglia out to the peripheral effectors. So somatic nerves do not work in this way, somatic motor nerves, but autonomic motor neurons do function in this way. So they leave the spinal cord, travel down an axon to an autonomic ganglia, synapse onto a second neuron, and that neuron leaves the autonomic ganglia and goes and synapses on the effector cell. Interneurons are mostly in the brain, the spinal cord, and those autonomic ganglia. They are connecting sensory and motor neurons, so they're responsible for the distribution of sensory information, for coordination of motor activity, and they're also greatly involved in those higher functions of memory, planning, and learning. Neurons perform all of the communication of our nervous system. They do all of the information processing and control all of our functions. So our neurons are the functional cells of the nervous system, specialized for communication, information processing. Neuroglia are supportive cells that preserve the physical and biochemical structure of neural tissue, and they are essential for the survival and function of neurons. So our neurons need to be in a specific environment, they need to be cared for, and the neuroglia are the cells that care for them. You'll find these pictures on page 396 and 397 in your textbook. So our neuroglia, we have, if you see, all right, so our neuroglia are the cells that support our neurons, and they have very important functions. You'll see that we're split right down the middle here. You'll find this on page 396 and 397 in your textbook. On the left-hand side, you'll see that we have our neuroglia in the peripheral nervous system. And on the right-hand side, we have neuroglia in the central nervous system. So you can see the peripheral nervous system has two different neuroglial cells, and the central nervous system has 4 different neuroglial cells. So we'll start with the peripheral nervous system. We have satellite cells, which surround neuron cell bodies within those autonomic ganglia that we were discussing, and they regulate oxygen and carbon dioxide as well as nutrient and neurotransmitter levels. So our nervous system has a really tight homeostatic state that it likes to be in, and so the satellite cells help to keep everything functioning really well in those autonomic ganglia. And then we have Schwann cells, and the Schwann cells wrap around the axons within the peripheral nervous system, and they myelinate, let's see here, so they myelinate the peripheral nervous system axons. So they participate in the repair process after injury. We're going to talk more about myelination in a few minutes, but it's very important that the Schwann cell is what allows for repair in the peripheral nervous system. We do not have Schwann cells in our central nervous system. Therefore, the nerves of our central nervous system have very limited, if any, ability to repair. So peripheral nervous system, we have satellite cells in the ganglia, and we have Schwann cells wrapping around the axons of the peripheral nerves. In the central nervous system, we have 4 different types of cells. So the first one on the left is the oligodendrocyte. Oligodendrocytes do myelinate the axons within the central nervous system, but they don't wrap all the way around the axons like the Schwann cells do, which is why they don't give that added ability to repair. Astrocytes maintain the blood-brain barrier. That's a very important thing to remember. We're going to talk more about the blood-brain barrier later, but the astrocytes maintain that blood-brain barrier, which is very protective for the brain. They also offer structural support, regulate ion, nutrient, and dissolved gas concentrations, absorb and recycle neurotransmitters, and form scar tissue after injury. Microglia are our phagocytic cells within the central nervous system, so they remove cellular debris, waste products, and pathogens. You certainly don't want to get an infection in your central nervous system, so in the event that some kind of pathogen does make it through the blood-brain barrier and infects your central nervous system, your microglia are there to go and clean them up. And then last, we have ependymal cells up on the top right. Ependymal cells line the ventricles, which are like large open spaces within the brain itself, and the central canal, which is a canal through the center of the spinal cord. And what they do is they secrete cerebrospinal fluid. So your brain and your spinal cord are completely surrounded and bathed in cerebrospinal fluid. And the ependymal cells are the ones that secrete or make this cerebrospinal fluid. Most axons in neurons within mammals are surrounded by a myelin sheath. And the myelin sheath is like a fatty, has fats and protein, and it's this protective layer, this covering for the neurons. Its function is to electrically insulate the axon and increase the speed of nerve impulse conduction. There you go. Increase the speed of nerve impulse conduction. So we're going to look at myelinated versus unmyelinated nerves. Myelinated nerves have nodes and internodes. So the internodes are the actual myelinated segments of the axon. And the nodes, which are also called the nodes of Ranvier, are gaps between the internodes. And this is if you were going to have a collateral branch off of an axon, they would branch at the nodes. We have 2 neuroglia that produce myelin sheaths, the Schwann cell in the peripheral nervous system and the oligodendrocytes in the central nervous system. Schwann cells begin to form sheaths around axons during fetal development, so very, very early. And what they do is they spiral around the axon. And the cytoplasm and the nucleus of the Schwann cell on the outside of that whole spiral forms what's called the neurolemma. The inner layer of that is the myelin sheath. So let's take a look here. The left-hand side, let's see, we're going to cut this picture down the middle. So the left-hand side is myelination with a Schwann cell. So you can see over here, the internode is the actual myelinated segment. So like that whole segment there, there's one internode, 2, 3, 4, 5, 6. We have 7 internodes here. And then the nodes are the areas in between the internodes. So let's see. So this would be a node here, well, a little bit below that, here, here, there. So the nodes are in between those internodes. If we start looking at this picture here, you can see that the yellow is the Schwann cell, and then it has this large nucleus in the cell itself. So the cell begins to wrap, so it's wrapping like this around the axon. The blue is the axon of the neuron. So it starts to wrap around the axon and forms this thick myelin sheath. So here you can see that whole dark spot is the myelin sheath, and that is from this fatty protein shell of the Schwann cell wrapping around the axon. So the axon is in the middle, and then the myelin sheath is around it, like that. The whole thing is the neurolemma. So the neurolemma, if we're looking at this picture, would be all around the outside. So you have the myelin sheath, which is directly on the axon, but then the neurolemma is like an additional membrane that surrounds the entire axon and includes the nucleus of the Schwann cell. Oops. Over on the other side, we are looking at unmyelinated axons. So this is a myelinated axon. Over here, we're looking at unmyelinated axons, and we do have those as well. So they have a Schwann cell, but instead of, you see on this side, the nucleus of the Schwann cell is off to the side and the axon is wrapped up. On this side, the nucleus is in the center, and then it's engulfing or encompassing multiple axons. You see all those little blue are the axons, and so they're all being held in place, but this Schwann cell has not wrapped around them, so it has not formed a neurolemma. So there's a difference between a myelinated cell or a myelinated neuron and an unmyelinated neuron. So unmyelinated neurons still have Schwann cells holding the axons together, but they are not forming a neurolemma around them. This is on page 400 in your textbook. So now we're finally going to talk about why is the neurolemma important. The neurolemma we only find around axons in the peripheral nervous system. And if an axon gets injured, the neurolemma can help in the regeneration of that nerve. It forms a tube that guides the regrowth of the axon. So as long as the neuron cell body is intact after injury, that neuron can often regenerate and repair, which is really important. If you get a bad, cut on your arm and they can reconnect everything together, you can have regeneration of neurons instead of losing all the sensory and motor control to your hand. The central nervous system does not have Schwann cells, therefore does not have neurolemma, and so they have a very limited regenerative capability. So we're going to go through some pictures of what happens with injury and regeneration within the peripheral nervous system. So step one, we've had an injury. You can see our injury has occurred right here. And we have what's called Wallerian degeneration, which is Wallerian, Wallerian generation, which is where so here you can see that the cell body is here, so off to this side, like that. So our injury was in the middle, so our cell body is still intact here, and the proximal neuron is intact, but this distal segment, the distal neuron, begins to degenerate. That's called Wallerian degeneration. Next, our Schwann cells are going to, so here are new Schwann cells, and they're going to grow toward the proximal portion of the neuron and form a sheath. For this, so here we have the distal axon is starting to grow into the region where the degeneration took place. And then you can see these purple cells are macrophages, which are cleaning up debris. So now we have this axon, or the myelin sheath, the neurolemma is intact again, and these Schwann cells are going to begin to grow around that neuron as it is sending off its new axon there. That's our axon bud. And so last, the axon is going to continue to grow until it is completely enclosed by Schwann cells and therefore regenerated. So that's really important that we can have this regenerative process within the peripheral nervous system. And again, I'll remind you, it is the Schwann cell that creates the neurolemma with myelinated axons that gives our peripheral nervous system the ability to regenerate. Oligodendrocytes are myelinating cells within the central nervous system. So a good test question could be, what could it be? Schwann cells are to the peripheral nervous system as blank are to the central nervous system, and the answer would be oligodendrocytes, because our Schwann cells myelinate the peripheral nervous system, oligodendrocytes myelinate the central nervous system. However, with their myelination, their cell bodies and their nuclei do not completely envelop the axon, so there is no neurolemma. In our central nervous system, we have gray matter and white matter. We see this in the brain and the spinal cord. Our white matter is composed of myelinated processes of many neurons in one place. Our gray matter is neuron cell bodies, dendrites, unmyelinated axons, axon terminals, and neuroglia. So the myelination gives nervous tissue a white color, and all of the unmyelinated pieces give that nervous tissue a gray color. Ganglia are masses of neuron cell bodies. The singular is a ganglion. They are surrounded by neuroglia, especially those satellite cells, and we find them in the peripheral nervous system. We do not have ganglia in the central nervous system. We have ganglia in the peripheral nervous system. All right, so now we're going to get into a little bit more physics of how signals are sent throughout the nervous system. So a transmembrane potential requires three different things. A concentration gradient of ions, so we have to have sodium on one side, potassium on the other side, and selectively permeable membrane in between. So we need the ability to keep sodium and potassium separate, and we need the ability to allow them to flow freely. Keeping them separate maintains a difference in charges across the membrane. And in a resting cell, resting membrane potential is negative 70 millivolts. With the negative 70, the negative means the inside of the cell is negative with respect to the extracellular fluid. So the fluid outside the cell has a more positive charge. The fluid inside the cell has a more negative charge. This would be a good time to watch Crash Course Neurology Part 2 because it would give you a nice little overview of transmembrane potential and what we're going to talk about. So across the membrane, the plasma membrane of the cell, we have chemical gradients, which would be the concentration of sodium versus the concentration of potassium. Remember that ions always want to move down their concentration gradients. And then we have electrical gradients, which are the separation of positive charges outside the cell and negative charges inside the cell. So we have a potential difference in electricity. So here is our transmembrane potential. In this picture, we are at resting membrane potential, which is negative 70 millivolts. So you can see we have all of these sodium ions outside the cell. So that's going to be giving us a positive charge outside the cell. And we have potassium ions are also positive, but inside the cell we have these large negatively charged proteins. So the overall charge inside the cell is negative because of these huge proteins. And the overall charge outside the cell is positive. So we have leak channels that are allowing some potassium to come out and leak channels allowing some sodium to come in, but overall we have this positive outside, negative inside. This is on page 403 of your text. An electrical current is a movement of charges to eliminate a potential difference. So when we have a positive charge outside and a negative charge inside, the current would move to eliminate those two and sort of make the charges equal on the inside the cell and the outside the cell. Resistance is the amount of current that a membrane can restrict. So we have a selectively permeable membrane, so it can restrict those electrical movements and it can keep that positive on one side and the negative on the other side, and that is through resistance. We can combine the electrical charge and the chemical gradients to form what's called the electrochemical gradient. So the electrochemical gradient for a particular ion is the sum of the chemical forces and the electrical forces acting on a specific ion across a plasma membrane. And this gives us a form of potential energy. Equilibrium potential is a transmembrane potential at which there is no net movement of a particular ion across the cell membrane. So we look at specific ions for this, not the total electrochemical gradient, but just the specific ion. So for potassium to be at equilibrium potential, the transmembrane potential would be negative 90 millivolts. And for sodium to be at equilibrium potential, the transmembrane potential would be positive 66 millivolts. So we're going to take a look at these. So here we're looking at potassium. This is on page 404 of your text. So the potassium at normal resting membrane potential, the chemical gradient of potassium wants to force potassium out of the cell, because you can see that there's a lot of potassium inside the cell and there's only a few potassium outside the cell. So chemically, potassium wants to go out of the cell. Electrically, potassium wants to enter the cell because there's a positive charge outside the cell, which you can see here, and there's a negative charge inside the cell, Potassium is a positively charged ion, so electrically potassium wants to go into the cell. So if we add the chemical gradient wanting to exit the cell along with the electrical gradient wanting to enter the cell, the net electrochemical gradient is a smaller force wanting to exit the cell. So our equilibrium potential would be where we have a small amount coming out and a small amount going in would be at negative 90 millivolts, because then there's a smaller chemical gradient trying to drive potassium out, and it has an equal electrical gradient trying to drive potassium in. Sodium is a little bit different. So at resting membrane potential of negative 70, there's a lot of sodium outside the cell and a few sodium inside the cell. So chemically, sodium wants to enter the cell. Then we are positive outside the cell and negative inside the cell, and sodium is a positively charged ion. So electrically, sodium wants to enter the cell. So when you add both the electrical and chemical gradients of sodium, you have a huge desire for sodium to enter that cell. So in order for the electrical and chemical gradients to be balanced, we would have to have a lot more sodium inside the cell. So the cell would be more positively charged on the inside. And so the electrical chemical gradient combined would be at the equilibrium potential would be at positive 66 millivolts. Because you've got more sodium coming into the cell, which is making the inside of the cell become more positive. In order for us to maintain our transmembrane potential, we need to have an active force that helps us. So our main active force is called the sodium-potassium ATPase, or sodium-potassium exchange pump. It is powered by ATP, so you must have ATP in order for the sodium-potassium exchange pump to work. And it carries 3 sodium out of the cell while transporting 2 potassium into the cell. So here's our cell. So we've got sodium coming out, 3 sodium coming out versus 2 potassium coming in. So our sodium-potassium exchange pump is pumping 3 sodium out of the cell while bringing 2 potassium into the cell. So this is working against the electrochemical gradient for sodium. It balances our passive forces of diffusion and maintains our resting membrane potential. Transmembrane potential can rise or fall in response to temporary changes in membrane permeability. So remember that our membranes are selectively permeable, which means that things can't just freely diffuse in or out. So when we open or close membrane channels, like a sodium channel, all of a sudden sodium would be allowed to rush into the cell, and that would change the transmembrane potential drastically. This is very, very important. Our transmembrane potential is determined by the membrane's permeability to sodium and potassium. So we have channels that bring sodium and potassium into and out of the cell. They cannot diffuse through the cell membrane, so they need to come through in channels. We have active channels and passive channels. Our passive channels are called leak channels, and those channels are always open, and so they're allowing sodium and potassium to come in and out of the cell. But there aren't a ton of them, so they can't make great changes in the transmembrane potential. Our active channels are gated channels, and they open and close in response to a stimuli. At resting potential, most of our gated channels are closed while our leak channels are open. So we have sodium and potassium leaking in and out of the cell, and the sodium-potassium exchange pump pumping sodium out of the cell and potassium into the cell. And so this is how we maintain the resting membrane potential, but those gated channels are how we change the transmembrane potential to create what our action potential. All of our plasma membranes, our cell membranes, produce electrical signals or action potentials by the movement of ions across the membrane. Transmembrane potential is particularly important to neurons because the action potential is how our nervous system communicates. Resting potential is the transmembrane potential of a resting cell. We've talked a lot about that. So a cell at rest has a resting potential of about -70 millivolts. A graded potential is a temporary localized change in the resting potential, but the effect doesn't go very far from where that change occurs. So it's sort of like a mini excitement, but then you settle back down, and it does not actually produce an action potential. But the action potential is an impulse that is propagated along the surface of the axon and does not diminish as it moves away from its source. So the graded potential diminishes the farther it gets from the source. The action potential is strong and it stays strong until it gets to the synapse. At the synapse, the synaptic activity is the action in response to an action potential that is then going to produce a graded activity in the postsynaptic cell. So the neurotransmitters get released from the presynaptic cell, such as acetylcholine at the neuromuscular junctions. Then the neurotransmitters bind to receptors on the postsynaptic plasma membrane and alter its permeability. Integration processing is the response of the postsynaptic cell. So the postsynaptic cell binds those neurotransmitters, and then it's either enough to produce an action potential within the second cell, or it's not, then it produces a low graded potential and it goes away. But these are the steps in creating and propagating an action potential. So here's our little overview in pictures. So over here we have our resting membrane potential. So this cell is just kind of hanging out. And then it's going to get a stimulus, and the stimulus will produce a graded potential. So the graded potential can be at the axon hillock, it can be in the body. So it can either kind of fizzle out and then nothing happens, or it can trigger an action potential. If it triggers the action potential gets propagated all the way down the axon and induces synaptic activity. So that synaptic activity would be the release of neurotransmitters from the presynaptic neuron, which will cross the synaptic cleft and bind to receptors on the postsynaptic cell. So we've already talked about this, that our ions cannot diffuse through our plasma membrane, so they must go through ion channels. When they're open, the ions are allowed to diffuse down their electrochemical gradient from areas of higher concentration to areas of lower concentration. So we have two types of ion channels, the leak channels and the gated channels. So our leak channels are always open, and their permeability is variable based on shape changes in response to local proteins. So even though they're always open, they may be less permeable or more permeable. And these are important in establishing our normal resting membrane potential because they've always got some ions flowing in and out. Our active channels or gated channels open and close in response to the stimulus. So we've covered this, but it doesn't hurt to say it twice. The presence of gated channels in the plasma membranes of neurons and muscle fibers is what gives them the property of excitability. Excitability is the ability to propagate an action potential. So in order for cells to be able to create an action potential or propagate an action potential, they must have a resting transmembrane potential and gated channels that allow you to quickly change that membrane potential. So that is the property of excitability. You'll find these next few pictures on page 406 in your textbook. So this first one, we have 3 different types of gated channels. So our chemically gated channels open in the presence of a specific chemical, like a neurotransmitter. So this is what it looks like at the neuromuscular junction, where you have acetylcholine coming in and binding to this receptor, and that binding allows the sodium gates to open and sodium to rush into the cell. It can also, the receptor can be for hormones or for specific ions. We find these on neuron cell bodies and on the dendrites of neurons, and these are used in the generation and conduction of action potentials. Voltage-gated ion channels respond to changes in transmembrane potential. So they're actually responding to the electrical charge, or the difference in electrical charges on the interior and exterior of the cell. So they have an activation gate, which opens it, and an inactivation gate, which closes it. So they open at a specific transmembrane potential, and then they close at a different transmembrane potential. These are characteristic of excitable membranes. So in order to have an excitable membrane, you must have voltage-gated channels. So you have to have a chemically gated channel to induce the action potential, and then a voltage-gated channels to propagate the action potential. So we find these in the axons of neurons and the sarcolemma of skeletal muscle and in cardiac muscle. So what this is showing is at resting membrane potential, the activation gate is closed, and so no ions are able to enter the cell. At threshold here, -60, so it's -55 to -60. I'm going to give you 2 different numbers throughout the rest of this lecture, and I'll go with either one. So around -60, you can see this activation gate has opened, and these sodium are allowed to rush in. And then when you get to +30, that is the deactivation, and so that is going to close the inactivation gate, and then that's not going to let any more sodium in. So this is it doesn't open until you get to -60, and it closes when you get to +30, so you can't change the resting membrane potential any more than that. Then we have mechanically gated channels, which respond to distortion of the plasma membrane. So if you were to press, here's an applied pressure. So if somebody were to, squeeze your arm really hard, that pressure would open mechanically gated channels and create an action potential. So we find these in touch, pressure, and vibration receptors. Graded potentials we talked a bit about, also called local potentials. These occur when the stimulus that's caused by the opening of that gate is not enough to stimulate an action potential. So therefore, they cannot spread far from the site of stimulation. Any stimulus that opens a gated channel produces a graded potential. A graded potential usually only affects the cell body and the dendrites, but usually does not trigger the action potential within the axon. So we're going to look at this in pictures. Here we're at resting membrane potential of negative 70 millivolts. Now we've got this chemically gated ion channel that allows some sodium to enter the cell. And that's going to bring us down to negative 60. And then we can have some more sodium coming in with this second chemically gated ion channel. But then the local current is going to start to decrease, and so it hasn't made enough to really trigger that action potential. So this is the initial site, and the farther we get away from the site, the less of an effect it has. So we end up out here going back to resting membrane potential. So a graded potential is a small change in the transmembrane potential that is not enough to trigger an action potential. So the farther you get from the initial stimulus, the smaller the change gets, and it'll sort of fizzle out, and you will eventually go back to resting membrane potential. This is called a graded depolarization. So depolarization is when you become less negative. You depolarize, and then repolarization is where you become more negative. So this is a graded depolarization. So again, a depolarization, you are becoming more positive or less negative. Repolarization, when that stimulus is removed, the transmembrane potential is going to return to normal. So repolarization is becoming less positive or more negative, going to resting membrane potential. And then hyperpolarization is an increase in negativity of the resting potential. So if your resting membrane potential is negative 70 millivolts, hyperpolarization would be like negative 80 or negative 90 millivolts. Hyperpolarization is usually brief and occurs after an action potential. During depolarization, sodium is coming into the cell. During repolarization, sodium and potassium are pumped out of the cell. So here's our graded potential. So we have our stimulus here, and then we get this little depolarization, but it does not depolarize to threshold. So threshold up here is where if you depolarize to threshold, then you're going to make your action potential. But a graded potential depolarizes some, but does not depolarize to threshold. So then you have a repolarization of that membrane. Hyperpolarization is where you become more negative, and then you'll go back up and repolarize or depolarize back to resting membrane potential. So again, that graded potential depolarizes the cell, but does not depolarize to threshold. So you end up just repolarizing and no action potential occurs. Graded potentials can be super helpful. They can trigger specific cell functions, like trigger a cell to release its products by exocytosis, or trigger a gland to release its secretions. You don't need a whole action potential in a gland in order to release secretions. A graded potential is sufficient. At the motor end plate, a graded potential is what allows the acetylcholine to be released into the synaptic cleft. When that happens, that can lead to an action potential in the postsynaptic cell. An action potential is a sequence of rapidly occurring events that decrease and eventually reverse the transmembrane potential and then restore it back to a resting state. So it begins the transmembrane potential begins negative, and then it goes positive, and then it comes back down to its normal resting negative state. And action potentials affect an entire excitable membrane. Once you stimulate an action potential, it's going to travel through the whole membrane. It is not going to stop. So it's an all or none principle. If a stimulus exceeds threshold, so that threshold is that negative 60, if it exceeds threshold, the action potential is going to be generated and it is going to be the same no matter how large the stimulus. So you either trigger an action potential or you don't. So here's our breakdown of action potentials. This is on page 410 of your textbook. So in this one, we are at resting membrane potential, and we have these voltage gated channels that are all closed. So step one, we're going to depolarize to threshold. So we have a current that's coming down the cell membrane, and it depolarizes us to threshold, that negative 60 millivolts. Once we depolarize, the sodium channels, the voltage gated sodium channels are going to open, and sodium is going to rush into the cell and rapidly depolarize that cell, bringing the transmembrane potential more and more positive. So the inner membrane of the cell is going to change from a negative polarization to a positive polarization. Step 3, when we get to positive 30 millivolts, that's going to trigger the sodium channel gates to close. So the inactivation gate is going to close on the sodium channel, and the activation gate is going to open on the potassium channel. So now we have no more sodium coming in, and we have potassium rushing out, and that begins our repolarization. As the potassium channels begin to close here, the resting membrane potential is going to, they'll start to close when we get back to resting membrane potential, and then while they're continuing to close, the membrane is going to hyperpolarize down to negative 90 millivolts, and then we'll come back to our normal resting potential as things start to return to normal. So a little review of what we just said. We started at resting membrane potential of negative 70 millivolts. A local current came that depolarized us to threshold, depolarized that membrane to threshold, which was negative 60 millivolts. That depolarization opened the sodium channel gates and allowed sodium to rush into the cell until the membrane potential reached positive 30 millivolts. Once we reached positive 30 millivolts, the sodium channel gate closed and the potassium channel gate opened. So the potassium started flowing out of the cell, which started to repolarize the cell. When we got back to resting membrane potential of negative 70, the potassium channels begin to close, but while they're in that process, additional potassium is leaking out. So we briefly hyperpolarize down to negative 90. But then the sodium-potassium exchange pump starts to work, and we quickly get back to our resting membrane potential of negative 70. During and after an active potential, action potential, we have what's called refractory periods. So the refractory period, we have two of them. The absolute refractory period is from when the sodium channels are open or are inactive. So you cannot possibly create another action potential because the sodium channels are already open or they are inactivated. So the absolute refractory period is shown here in pink. So it is from when you reach threshold, because now all the sodium gates are open, until here, now up at number 3, the sodium gates are inactive. The inactivation gates are closed and the potassium gates are open, so we are starting to repolarize. So this whole period here, is the absolute refractory period. So there's nothing you could do to create an additional action potential during the absolute refractory period. Following the absolute refractory period, we have what's called the relative refractory period. So during that time, the membrane potential is almost normal. It goes from normal to hyperpolarized, and then it comes back up to normal. And while it's in this phase, a very large stimulus can initiate a second action potential. So you would have to, when you're hyperpolarized, it takes more to get to -60 from -90 than to get to -60 from -70. So when you're in that hyperpolarized state, a larger stimulus can create an additional action potential. During the absolute refractory period, nothing can happen to create an additional action potential because the sodium channel gates are already open or they are inactivated. The sodium-potassium exchange pump is maintaining the concentration of sodium and potassium over time, and it requires ATP. Without ATP, our neurons stop functioning because in order to create and propagate an action potential, you have to have a difference in transmembrane potential that is more positive on the outside due to the sodium concentration and more negative on the inside due to the negatively charged proteins. So, and remember that we have 2 potassium being pumped out and 3 I do that every time. 2 potassium being pumped in, 3 sodium being pumped out. All right, so potassium in, sodium out. I feel like a kindergartner trying to draw on this thing, but it is what it is. Okay, so 1 ATP pumps 2 potassium into the cell and 3 sodium out of the cell. I'm going to encourage you to spend a fair amount of time going over all of that in your studies this week. That is the more challenging bit of material for this week, is what is resting membrane potential, what is threshold potential, what is a graded potential versus an action potential. Those are all very important steps in learning the nervous system function and how we can continue to feel, sense, smell, move. All of that stuff is based on the creation and propagation of action potentials. So to propagate an action potential means to relay an action potential from one location to another location. So if we want to wiggle our big toe, our brain has to send information all the way down the nerves to our big toe to get our muscles to contract to move our toe. And that sending of information is called propagation. So we have 2 styles of propagation, continuous and saltatory. Continuous propagation is along unmyelinated axons, and what happens there is the action potential has to affect every portion of the membrane surface. So I would typically draw this on the board. So here's one, let's see, there's one neuron, and then this is going to be our second neuron, and our second neuron has Schwann cells wrapped around it. All right, so our first neuron, in order for an action potential to be generated, the action potential has to touch every single piece of that cell membrane in order to continue down the axon. Saltatory conduction is much faster and it propagates the action potential between successive nodes of a myelinated axon. So remember, the nodes are these parts that are in between the internodes. So what this does is, with myelinated axons, the signal jumps from node to node. Instead of having to touch every single part, like in the unmyelinated axon, it just quickly depolarizes the adjacent node, and so it sends information much more quickly. So that myelination is very important to the speed of propagation of action potentials. So I basically just explained this slide that our current is jumping from node to node or leaping, and so it's faster and it utilizes less energy than continuous propagation. The speed of the propagation of an action potential depends on the myelination status. We just talked about that. Myelinated is faster, unmyelinated is much slower. The diameter of the axons of the nerve. So the larger the diameter, the faster the propagation, because there's more cytoplasm or more axoplasm and less resistance to flow. So the larger diameter, faster propagation. And then temperature. The temperature is faster when it's warm and colder or slower when it's cold. And I can tell you this by experience. I did the Tough Mudder a few years ago, and it was 95 degrees outside. It was really not a great day to be doing the Tough Mudder. But we got to this, we were probably 75% of the way through the race. So we had gone, say, 9 miles at this point, and we had to go down a slide into ice water. And there's chicken wire over the slide, so you have to lay down and you have to go and submerge in the ice water to get under the chicken wire to be able to get out. And you ended up having to go about 5 feet through this big trough of ice water, and then climb over a wall, and then go another 5 feet through water, and then climb over a wall to get out of the obstacle. And I was on fire. Literally, it was 95 degrees out, and I had just run 9 miles and done, I don't know, 35 obstacles. And I entered that water, and I felt myself immediately starting to slow down. And when I got to that wall, it was all I could do to climb over the wall. And halfway up the climb, I fell into the next bin of water. And I literally did not think I was going to be able to get myself out of that second portion of water. It was like my brain was screaming at my body to move, and my body was not listening. I could not force my muscles to contract. It was the craziest feeling I've ever had. So that cooling down the body decreases the speed of propagation of an action potential. And it can be very dangerous. So when people go through a lake into freezing cold water in the winter when they're out ice fishing, your body shuts down so fast that you do not have the ability to swim up and save yourself. So be careful if you're an ice fisherman. We have 3 different groups of axons: type A fibers, type B fibers, and type C fibers. These groups are classified by their diameter of the axon, the myelination status, and the speed at which the action potential propagates. Type A fibers are myelinated. They are large diameter, they are high speed, they carry very rapid information. So information about position, balance, touch, motor commands are all type A myelinated fibers. I thought I'd change this in here. You can cross out express mail and write text message. So type A fibers would be like sending a text message. These are a little bit, that is a backwards E. Apparently we're almost done with this lecture. This is what happens at the end. So a text message would be the super fast type A myelinated fiber. Type B fibers are also myelinated, but their diameter is smaller, so they transmit at a lower speed than the type A fibers. So more of our intermediate signals. And this would be more like sending an email. So if you want to get somebody's attention right away, you're going to text them. If you want them to look at something within a couple hours, you might send them an email. And then type C fibers are unmyelinated, small diameter, super slow speed. They carry slower information like involuntary muscle, so smooth muscle controlled, glandular control, things like that that don't need to happen quickly. And this would be more like snail mail, like you're mailing a letter to somebody. So text message, you want them to see it immediately, type A fibers. Email, type B fibers, like I still want you to see it, but it doesn't have to be immediate. And then type C fibers is like, I'm going to mail you a letter, and in a couple of days, you're going to tell that gland to secrete. It doesn't really take a couple of days, but it's much, much slower. All right, this would be a good spot for you to watch the Crash Course Part 3, Crash Course Neuro Part 3. We're going to talk about what happens at the synapse. So action potentials are transmitted from one neuron to the next, or from a neuron to another postsynaptic cell. So the neuron carrying the information is the presynaptic neuron. Then the cell that is receiving the information is the postsynaptic neuron or a postsynaptic cell. And those two cells are separated by what's called a synapse. We have electrical synapses, which have direct physical contact between cells, and chemical synapses using neurotransmitters to send chemicals from one cell to the next. Electrical synapses, the pre- and postsynaptic cells, are locked together at what's called a gap junction. Gap junctions allow for rapid transfer of ions between two cells, and they create what's called a common membrane effect, which means that you can have a continuous propagation of information through the entire system. So areas of the brain, the eye, the ciliary ganglia, these are all connected by gap junctions, rapid communication between two cells. Chemical synapses are found in most synapses between two neurons and in all synapses between neurons and other cells. So our chemical synapses are much, much, much more common than electrical synapses, but we like to tell you that there are two. So synapses are at the chemical synapses, cells are not in direct contact with one another. They are separated by what's called the synaptic cleft. At a chemical synapse, the action potential may or may not be propagated to the postsynaptic cell, depending on the amount of the neurotransmitter released and the sensitivity of the postsynaptic cell. That is important. Let's do it in yellow. The sensitivity of the postsynaptic cell is important. So if you are a nerve and you are releasing acetylcholine and the postsynaptic cell does not have acetylcholine receptors, then you cannot affect the postsynaptic cell. So the postsynaptic cell has to be sensitive to the neurotransmitter that you're releasing, and it has to have open receptors. If the receptors are already bound to acetylcholine, then the release of additional acetylcholine is not going to affect the postsynaptic cell. We have two classes of neurotransmitters: excitatory neurotransmitters, which cause depolarization of the postsynaptic membrane and propagate action potentials, or stimulate the creation of an action potential in the postsynaptic cell. And inhibitory neurotransmitters, which hyperpolarize the postsynaptic membrane and make it harder to create an action potential. So sometimes you don't want a cell to be stimulated, so you can hyperpolarize it and make it more difficult to stimulate that cell. And here again, the effect of the neurotransmitter on the postsynaptic membrane depends on the property of the receptor, not on the nature of the neurotransmitter. So the property of the receptor is what we were just talking about, that if the receptor is an acetylcholine receptor and the neurotransmitter is acetylcholine, then that's going to affect that cell. If the receptor is an acetylcholine receptor and the neurotransmitter is epinephrine, the epinephrine is not going to affect the postsynaptic cell because it only has receptors for acetylcholine. Cholinergic synapses are synapses that release acetylcholine. So this is the main one that we've talked about. It is found in all neuromuscular junctions and involved in skeletal muscle, in many synapses within the central nervous system, at all neuron-to-neuron synapses in the peripheral nervous system, and at all neuromuscular and neuroglandular junctions within the parasympathetic division of the autonomic nervous system. So they are by far the most common synapse. It is the most common neurotransmitter, are cholinergic, utilizing acetylcholine. So here's what happens at our cholinergic synapse. So first we have the action potential arriving. So this is coming down the axon and down the telodendria into the synaptic knob. So remember, this whole area is the synaptic knob. Then that arriving potential opens these calcium gates and allows calcium to enter the synaptic knob. That calcium is what triggers the release of acetylcholine through exocytosis. So first thing, so in the presynaptic neuron, the action potential is coming down the axolemma, stimulates the opening of calcium channels, and then the calcium stimulates the release of acetylcholine through exocytosis. Then the acetylcholine is going to cross the synaptic cleft and bind to the receptor on the postsynaptic membrane. So this is a chemically gated ion channel because the chemical that it's looking for is acetylcholine versus the voltage gated, which changes in response to the transmembrane potential. So chemically gated ion channel, the acetylcholine binds, and that allows sodium to rush into the postsynaptic membrane. That sodium rushing in can cause a action potential in the postsynaptic membrane, or it can cause a graded potential in the postsynaptic membrane. Additional acetylcholine is then broken down by acetylcholine esterase, and the choline is recycled into the synaptic knob and utilized with acetyl-CoA up here to create additional acetylcholine. So the parts get broken down and recycled to create additional neurotransmitters. There is a delay at the synapse of 0.2 to 0.5 milliseconds, which might feel like nothing, but that can add up if you have 100 synapses that you have to get through in order to transfer information. So that synapse is between the arrival of the action potential at the synaptic knob and the acetylcholine binding to the receptor on the postsynaptic membrane. So that's what that delay is, the calcium entering the knob releasing the acetylcholine crossing the cleft, and then attaching to the receptor on the postsynaptic membrane. The fewer synapses you have, the faster your action potentials are going to be transmitted. Some reflexes only involve one synapse, so it is super fast. Like if you're sitting and I test your patellar reflex, I hit your knee, your foot moves instantaneously because there's one synapse in that link. Synaptic fatigue can occur when neurotransmitters are not being recycled fast enough to meet the demands of the intense stimuli. So if there are many action potentials coming down and you're not able to bring that choline back and create more acetylcholine, then that synapse is non-functional until the acetylcholine is replenished and can be released again. So it doesn't matter how many signals you're getting. If you run out of neurotransmitter, then you can't affect a postsynaptic membrane until you create more neurotransmitter. A neurotransmitter is a chemical compound released by one neuron to affect the transmembrane potential of another. So we've been talking about acetylcholine. There are other ones. There's some charts in a couple of slides that are going to tell you what the main neurotransmitters are, but that's a neurotransmitter. A neuromodulator is a compound released by a neuron that adjusts the sensitivity of another neuron to specific neurotransmitters. So we could decrease the sensitivity of a second neuron to a neurotransmitter or increase the sensitivity. So that's a neuromodulator, which is a compound released by the presynaptic neuron or released by a neuron to adjust the sensitivities of another neuron. So our action potentials are transmitted at a chemical synapse. You have the synaptic terminal releases the neurotransmitter, and like the acetylcholine, and that crosses the synaptic cleft and binds to the postsynaptic plasma membrane. That binding produces a temporary localized change in permeability or function of the postsynaptic cell. So in the neuromuscular junction, the acetylcholine created a temporary opening of sodium channels, allowing sodium to rush into the sarcolemma. Those changes affect the cell depending on the nature and number of stimulated receptors. So if you have one acetylcholine that affects one receptor that opens one sodium gate, that's going to create a small graded potential. But if you have 100, or I don't actually know how many there are, maybe there's thousands of acetylcholine receptors that all bind acetylcholine at once, and it opens 1,000 sodium gates, you could see how different that would be on the transmembrane potential of the postsynaptic cell. Many drugs affect the nervous system by stimulating receptors that respond to neurotransmitters. So they can have complex effects on perception, on motor control, and emotional states. So one example is that morphine binds to the postsynaptic receptors and inhibits what's called substance P. Substance P transmits pain signals, P for pain. So when the morphine binds to those receptors and inhibits substance P, then the pain signals cannot be transmitted. So that's how morphine helps to relieve pain. You can go on YouTube and look up opioid drugs mechanism of action, and it'll show you, there's a video on there that shows you sort of how opioids work to interrupt those pain signals. Neurotransmitters and neuromodulators work directly on membrane channels, like acetylcholine, glutamate, and aspartate are going to bind to membrane channels and allow them to open. They act indirectly via G proteins utilizing a second messenger. So that we're going to get into a lot later on in A&P2, but a G protein is a protein within a cell membrane that has a receptor on the outside of the membrane, and then it can stimulate a second messenger inside the membrane. So it can create a reaction within the cell by binding to something outside of the cell. So epinephrine, norepinephrine, dopamine, histamine, and GABA all utilize a G protein, which is an enzyme protein complex with a membrane receptor that is within the cell membrane. And then indirectly using second messengers. So like lipid-soluble gases, they can diffuse directly through the cell membrane and then trigger an enzymatic reaction within the cell without having to bind to the cell membrane at all. So here's our direct effect. This should look exactly like what we've been doing. So we have a chemically gated ion channel here. It's closed. It binds to the chemical, acetylcholine in this case, opens the channel, allows sodium to rush into the cell. So that's a direct effect, ionotropic, chemically gated ion channel. Now here we have our G protein. So how this works is you have the receptor here with a G protein bound to it, and that G protein is inactive, which means nothing's happening in the cell, and this is all inactive. So when you have, sorry, all right, that was an accident. When you have a neurotransmitter that binds to that receptor, the G protein is then going to stimulate enzymes within the cell to create a process that will go and open an ion channel within the membrane. So that's that the G protein. So go back and look at that again. I'm sorry, I accidentally flipped forward with the slide. This system does not allow me to flip back and speak about it again. And then our last are indirect effect using intracellular enzymes. We have, so here we've got nitric oxide, which is diffusing through the plasma membrane, and once it gets into the cell, it activates enzymes that produce a second messenger, and the second messenger goes and opens the channel in the membrane. So this is sort of how those G proteins work, by activating enzymes within the cell, but this one is showing you the intracellular enzymes being activated by the diffusion of nitric oxide through the cell membrane. The G protein requires, it has a receptor that receives the neurotransmitter, that activates the G protein, and then the G protein activates the enzymes within the cell. All right, so the next few slides here are neurotransmitters of note. Acetylcholine is the main neurotransmitter. It's the one that we talk about the most. So it's the one that I would want you to know. Please do not try to know the chemical structure of it. We know that it is direct, binding to chemically gated ion channels. And we talked about where they're found in the central nervous system, in the peripheral nervous system. It is the best known and most studied neurotransmitter. So I do want you to know just the most of this stuff about acetylcholine. The rest of these we're going to learn some more about as we go along, but we could teach an entire course on neurotransmitters. So in this portion of our class, I do not expect you to learn all of these different neurotransmitters. So if you are super interested, awesome. You can do a lot more research on this. Like serotonin is for important in emotional states and moods. So selective serotonin reuptake inhibitors are affecting the transmission and reuptake of serotonin. There's all kinds of really interesting stuff you can go into with these, but for now I'm going to let you do with this what you may. So learn about acetylcholine, and if you are feeling adventurous, then by all means go learn about the rest. But if you are not feeling adventurous, then I'm not going to require that you know all of this information about all of these neurotransmitters. So I just get a good grip on acetylcholine. All right, so we're going to talk about processing information. At the simplest level, you've got a neuron that has a whole bunch of dendrites, and those dendrites are receiving information all the time. So they're simultaneously receiving information from multiple different neurons. Some of the neurotransmitters coming in are excitatory, and some of them are inhibitory. So it processes all of the excitatory and all of the inhibitory information, and then that combination has a net effect on the axon hillock. The stimulation of the axon hillock is what determines if an action potential is actually going to be generated or not. Graded potentials develop in a postsynaptic cell in response to neurotransmitters. We have two types of postsynaptic potentials. So excitatory postsynaptic potential is a graded depolarization of the postsynaptic membrane. So depolarization. Excitatory is going to depolarize the postsynaptic membrane. Inhibitory is going to hyperpolarize the postsynaptic membrane. Remember, it's inhibiting it. It's making it more difficult to create an action potential in that postsynaptic cell. So it is inhibitory, and it hyperpolarizes the postsynaptic membrane. A neuron that receives many inhibitory postsynaptic potentials is inhibited from producing an action potential. So if there's a whole bunch of inhibitory postsynaptic potentials happening, then that neuron is not going to form an action potential because the stimulation required to reach threshold is increased. So if you bring that resting membrane potential down to -90 or lower, then it is much harder to create an action potential to get it up to -60 threshold potential, to which it's going to then create an action potential. The term summation is through the combination of multiple excitatory and or inhibitory postsynaptic potentials. So one excitatory postsynaptic potential is not enough to trigger an action potential in the postsynaptic cell. So you need to combine all of the potentials coming in to decide whether or not you can actually trigger an action potential. And there's two different ways that that can happen. Spatial summation is where you have multiple stimuli in different locations. The cumulative effect on the transmembrane potential of those multiple stimuli in different locations is what decides if you're going to trigger an action potential or not. So on this slide, you can see that we've got one potential coming in here, one coming in here, and so the combination of the two is going to either trigger an action potential or not. Temporal summation is where you have one region that's getting rapidly repeated stimuli. So the first time up here, the first stimulus, you can see that there's this little mini graded potential in pink. The second stimulus, you've got a larger potential, but you haven't quite gotten to threshold. The third stimulus, you've made enough of an potential to create and propagate that action potential. A neuron becomes facilitated when the excitatory postsynaptic potentials accumulate, and it raises the transmembrane potential closer to threshold, which allows a smaller stimulus to trigger an action potential. So a facilitated, if you think about, like if you sit too long and your leg falls asleep and it starts to wake back up, and all of a sudden all you have to do is brush your leg and it turns on all of your neurons and it hurts really badly when your leg is asleep. Those neurons are all facilitated. They're all close to threshold, and it takes a very little stimulus to fire all of those neurons. So here we've got our excitatory postsynaptic potentials, which are depolarizing, and inhibitory postsynaptic potentials, which are hyperpolarizing. And so if we have multiple stimuli coming in at the same time, some of them are excitatory, some of them are inhibitory. Those ones might cancel each other out. So it depends on if you have a more excitatory, you're going to create an action potential. If you have more inhibitory potentials coming in, then you're going to block the action potential. This is on page 426 of your text. So I know this was a lot of information today. This is our introduction to the nervous system. Over the next 3 weeks, we're going to cover the brain and spinal cord, the anatomy of each, how they function, how they transmit signals, the peripheral nervous system, so how we receive sensory information and how our motor commands are issued and carried through. We're going to look at reflexes of what happens when you touch a hot stove and how that differs from what happens when you step on a tack. And then we're going to talk about how information is organized within your spinal cord and all that kind of amazing stuff. The nervous system, when I was in school, I had 12 months of anatomy that was 6 hours of lecture and 6 hours of cadaver dissection every week. And then I also had a neuroanatomy class, which was 4 hours of lecture and 4 hours of just brain and spinal cord dissection every week for, I believe it was 9 months. So trying to teach you everything in 4 weeks is a Herculean task, but I'm going to try to teach you the basics and get you so that you can get excited about it and maybe go and further your education in the nervous system. So have a wonderful week, and I will see you back next week.